systems for hydrogen ion concentration in the blood to ensure they are detailed, comprehensive, and interconnected, while also providing a summary table and mnemonic for quick recall. Regulatory systems for hydrogen ion concentration blood. Quick summary table, key role, location. Mechanism, clinical significance. Maintains pH of 7.4. CO2 excretion via lungs, Brentsev sample 3A. Carbonic acid-bicarbonate maintains pH of 7.4. Blood, ECF 20.1 ratio of bicarbonate to carbonic acid. CO2 excretion via lungs. Kidneys regulate HCO3, first line of defense in ECF pH regulation. Hemoglobin buffer maintains acid-base via chloride shift RBCs, exchange of chloride and bicarbonate ions depending on O2 levels, facilitates oxygen delivery and CO2 transport. Plasma protein buffer adjusts hydrogen ion concentration. Plasma proteins bind or release hydrogen ions, liver involvement in protein synthesis, balances hydrogen ion concentration in plasma. Phosphate buffer neutralizes excess H+ ions, cells, body fluids, kidneys, converts strong acids, bases into weaker ones, especially in renal tubular fluid. Important in urinary pH regulation. Common mnemonic for quick recall. CATs help protect patients. Carbonic acid-bicarbonate, hemoglobin, plasma protein, phosphate. Detailed explanation. 1. Buffer systems overview. Buffers are the fastest acting regulatory system, providing immediate protection against pH changes by absorbing or releasing hydrogen ions, H+. Their function keeps the pH stable, ensuring homeostasis despite fluctuations in acid or base levels. 2. Primary buffer systems in extracellular fluid A. Carbonic acid-bicarbonate system. Role maintains a pH of 7.4 with a 20:1 ratio of bicarbonate HCO3 to carbonic acid, H2CO3. Mechanism, 1. Carbonic acid formation. Combines CO2 and H2O to form H2CO3 via carbonic anhydrase. 2. Regulation by lungs. CO2 excretion affects carbonic acid levels. 3. Kidney function. Adjusts bicarbonate levels by retention or excretion. Significance. Primary buffer of the ECF, critical in conditions like respiratory or metabolic acidosis alkalosis. B. Hemoglobin buffer system. Role. Maintains acid-base balance through the chloride shift in red blood cells. Mechanism, 1. Chloride exchange. Chloride ions move into out of RBCs based on oxygen levels. 2. Compensation. Each chloride ion movement is counterbalanced by a bicarbonate ion. Significance, supports gas transport and acid-base equilibrium. C. Plasma protein buffer system. Role, modulates pH by attracting or releasing hydrogen ions. Mechanism, plasma proteins, particularly albumin, act as weak acids or bases. Liver function, synthesizes proteins that adapt based on pH needs. Significance, vital in plasma pH regulation in metabolic conditions. D. Phosphate buffer system. Role, neutralizes excess H+ ions, especially in renal tubular fluid and intracellular fluid. Mechanism, 1. Converts strong acids, HCl, to weaker acids, H2PO4. 2. Balances H+ concentration in the kidneys. Significance, plays a major role in urinary pH maintenance. 3. Buffer limitations. Buffers are consumed during reactions, making the body vulnerable to continued pH imbalances until they are replenished. Underlying cause, acid-base imbalances must be addressed at the root cause for effective resolution. Clinical implications. 1. Respiratory acidosis, caused by hypoventilation, leading to CO2 retention, managed by increased bicarbonate buffering and renal compensation. 2. Metabolic alkalosis, often due to excessive bicarbonate or loss of H+, requires careful management of the bicarbonate system. 3. Renal tubular acidosis, indicates a defect in the kidney's ability to excrete H+ or reabsorb HCO3.